So good afternoon, everyone. We're going to go ahead and get started. Um, I'm thrilled to introduce today's Grand Round speaker, one of our own, Jeff Ishizuka, who's been with us for now 6 years, something like that, 5.5 years. Yes. So Jeff is an assistant professor in oncology. He also has an appointment in pathology and immunobiology. He works in our group. He takes care of patients with melanoma and other kinds of skin cancers. So Jeff has a very illustrious past. He came to us from Harvard University. He did his residency and fellowship up there in Boston at the Brigham, and then fellowship at the Dana-Farber Cancer Institute. His PhD comes from Oxford University, where he studied CD8 positive T cells and immunodominance and cross-reactivity. He then did part of his PhD, I think, at the NIH, and somewhere along the line, he became a Rhodes Scholar. Is that correct? The Oxford part. Anyway, all good. We were so happy when Jeff agreed to come here to Yale to join our group. He has become our go-to person for all sorts of insights in immunobiology. If you have experiments that you're having difficulty interpreting, I'm strongly recommending going to Jeff. So without further ado, we'll let Jeff get started. He's going to be talking to us today about targeting of tumor inflammatory states. So thank you so much, Jeff.
So thanks so much for the opportunity to speak today. These are my disclosures. None of them speaks particularly to anything I'll be talking about today. So I thought I'd actually start with kind of the origins of the lab. So our lab works at the interface of innate immunobiology, synthetic biology, and functional and single-cell genomics. However, we got our start working with Nick Haining and Rob Manguso at the Broad Institute in Dana-Farber in the days that they were doing some of the first in vivo CRISPR screens, kind of knocking genes out in transplantable tumor models and planting them under increasing immune selective pressure and looking for gene knockouts that specifically sensitize to immunotherapies. And one of the interesting things when they did these experiments is that a number of the hits they got back actually had to do with antiviral sensing pathways, kind of viral defense pathways in tumors. And a lot of this has been borne out in subsequent publications. But for me, we ended up working a lot on this gene, which is ADAR1. It's an adenosine deaminase that acts on double-stranded RNA and catalyzes the conversion of adenosine to inosine in double-stranded RNA species. So as it turns out, all of our cells are capable of making double-stranded RNA, whether from hairpin structures, from antisense species, or the expression of endogenous retroelements from various parts of the genome. And what ADARs do is they take these, or ADAR1 does, is it takes this double-stranded RNA and prevents cytosolic viral sensors like MDA5, RIG-I, and protein kinase R from being triggered. So in doing so, it prevents autoinflammation, type 1 interferon, translational arrest, and apoptosis. And what we had found is that if you knock ADAR out in tumor cells potentially being particularly vulnerable to this phenotype due to dysregulated chromatin states, that you can significantly remodel the tumor microenvironment. You can repolarize all of your suppressive myeloid cells to become inflammatory monocytes. You can drive CD8 T cell expansion and kind of activated phenotypes. And we were not the only ones to identify the antiviral sensing axis as a major driver of tumor inflammatory state. So whether it was looking at ligand abundance, and this is shown in studies including CDK4/6 inhibition, where they showed that with CDK4/6 inhibition, you de-repress endogenous retroelements, whether it's work done here at Yale by Chen Yan's group studying KDM5B, and again, looking at the expression of endogenous retroviruses, whether it's RNA editing. We had studied ADA-I editing, but Stephanie Helene here at Yale has looked at M6A RNA editing. Other forms of RNA editing can also control kind of tumor inflammation or inflammatory states. Ligand localization and sensing. This could be, micronuclei formation, or as Megan King studies here, DNA bridges, or finally signal integration, the downstream signals from interferon and, viral sensing pathways, which can drive either resistance, as studied by Andy Min at Penn, or kind of activated dendritic cell and other innate immune states. This is proven to be, again and again, one of the major drivers of tumor response and tumor inflammatory states that has come up. And as we started the lab here at Yale, it occurred to us that antiviral sensing may be one major driver of inflammatory states, but we don't really understand broadly what the other drivers of inflammatory state are, how you arrive at the particular inflammatory states in tumors that we observe in human tumors, or really, frankly, a full understanding of what those states are. So, here I'm showing the histologic immunotypes, immune deserts that lack immune cell infiltration, immune excluded tumors where the immune cells are pushed to the periphery, and inflamed tumors where CD8 T cells and other species kind of diffusely infiltrate tumors. And we know that very broadly, the former two may have a less correlation with immunotherapy response, and the latter may have a stronger correlation with immunotherapy response. But really a full depiction of what the variety of states are. Perhaps Max Krummel was here recently. The archetypes that he suggested are somewhat better than these histologic immunotypes. But I think it's fair to say we don't fully understand the variety and diversity of inflammatory states that tumors can adopt. Beneath that, even less, I would argue, do we understand the mechanisms by which these states emerge. And so we think that these are largely likely to be innate immune mechanisms, and that there is perhaps more back and forth between the immune system, the innate immune system, and early tumorigenesis than is currently recognized. But whatever it may be, that they're not very well understood. What you can see is that if you kind of squint, there are other evolutionary priorities for the immune system that reflect somewhat into these states. So if you look at deserts and shutdown states, they have some, at least surface correlation with the need to prevent autoimmunity and kind of shut down autoinflammatory reactions. In the excluded tumors, the need to heal wounds with, CAF-rich and macrophage-rich states. And then in the inflamed states, the need to protect against pathogens, and particularly intracellular pathogens and viruses. So within the lab, we became really interested in this question broadly: what are the core drivers of inflammatory states in tumors? And how do tumors trigger and manipulate the innate immune system? Ultimately, being interested in using these insights to develop better therapies. This kind of frames the lab goals, which are to define and target tumor inflammatory pathways to enhance immunity. And really, that means three things to us. The first is that we work to develop new tools and technologies to dissect tumor microenvironment inflammation. And the first thing that came to us is that it wasn't completely clear that the set of tools that we had was adequate to this task. The next is that we try to define and understand the regulatory biology of tumor inflammatory processes. This could be traditional mechanistic immunobiology, or this could be the application of CRISPR screens and the tools that we've developed. And the final thing is we try to advance some of this work towards the clinic and develop new therapies. So time permitting, I'll try to get through 3 stories today that all have their origin in new tools that we've developed or applied to understand tumor inflammatory states. And the first is really understanding double-stranded RNA sensing and cytosolic double-stranded RNA sensing within tumor cells. And here we started with the idea that there isn't really a set of tools that allow us to track double-stranded RNA in live cells and kind of continuously image specifically cytosolic double-stranded RNA, which is a type of double-stranded RNA that we've seen to remodel the tumor microenvironment in ADAR null tumors. And so we thought about what would design principles for an optimal sensor for double-stranded RNA look like. We thought 3 things. First, that it would be highly specific for double-stranded RNA recognition. Second, that it would have high sensitivity to double-stranded RNA without sequence or really high size specificity. And finally, that it would have a small size so that it could freely diffuse into subcellular compartments without disrupting native signaling. And we realized that such a thing already existed, although it had been largely studied within the plant literature. And basically, it's based on this protein B2, which is a derivative of the flock house virus and binds to double-stranded RNA. And so the way that it has been used and the way that we've used it is that if you create a split fluorophore, so half of a fluorophore is attached to 2 separate B2 proteins. Where there's double-stranded RNA in the cell, the B2 proteins sit down next to each other and reconstitute the fluorophore, and you can track double-stranded RNA abundance as well as subcellular localization. And to test this, we used a rhodamine-labeled poly-IC, which is a synthetic double-stranded RNA mimetic that's used frequently to study double-stranded RNA in the lab. And we transfected cells, and you can see kind of on the right in the rhodamine-labeled poly-IC, you can visualize the poly-IC, and that you get a really nice overlay with the B2 split fluorophore, suggesting that this is working to kind of detect double-stranded RNA. Interestingly, you can also see overlap, although not complete overlap, with 8R1, this other double-stranded RNA editing enzyme that I mentioned previously, suggesting that there's some diversity in the condensates and the species that kind of co-localize in these cells. So to validate that our sensor is truly a double-stranded RNA sensor, doesn't recognize single-stranded RNA and does recognize different sizes of double-stranded RNA in mammalian cells, we pursued electrophoretic mobility shift assays in collaboration with Anna Pyle's lab. And the way that you read this is that you titrate in different amounts of your B2 protein, and you see whether the free double-stranded RNA on the left or the single-stranded RNA on the right shifts. Right? And if it shifts, you can tell that there's binding happening. And as you see in the double-stranded RNA assay, you see shifts for multiple sizes of double-stranded RNA, particularly at the higher concentrations of B2 protein. And on the right, you don't see too much of a shift at all, suggesting that this is, in fact, pretty specific and sensitive for a range of double-stranded RNAs, but not single-stranded RNA. Okay? So with this tool in hand, we said, okay, we can start to visualize the dynamics of double-stranded RNA and cytosolic double-stranded RNA in cells. And so here, what we have done is, again, use rhodamine-labeled poly-IC in melanoma cells, as well as the B2 reporter. And we've also labeled the mitochondria. So a lot of the double-stranded RNA sensors live on the mitochondria, MAVs and MDA5 and RIG-I. And you can see where you see it turn white is actually where there's overlay between the green and the rhodamine-labeled poly-IC. And you can visualize these interacting with the mitochondrial membrane. See if I can get this to go. Yes. Furthermore, we can visualize the networks of double-stranded RNA that form in 8R null cells, cells lacking the RNA editing enzyme 8R1, following stimulation with interferon. So you can see these kind of reticular networks of double-stranded RNA in the knockout cells, but not in the control cells, suggesting that we can really use this as a tool to monitor the dynamics we're most interested in. So having validated this B2 reporter in mammalian cells, we wondered whether we could use it to actually understand and identify novel regulators of double-stranded RNA sensing. To do this, we combined two methods. The first is a mass spec pull-down method, where we use a tag on the B2 protein to pull down all the double-stranded RNA interacting proteins throughout the cell. We perform mass spectrometry, and we kind of looked at which proteins interact with double-stranded RNA. We combine that with a genome-wide CRISPR screen that we performed in vitro, looking at sensitization of tumor cells to poly-IC-induced stress. So basically, what genes, when deleted, actually sensitize tumors to dying following stimulation with poly-IC. And these are the results of these two experiments. On the left, you see the set of proteins that we identified as interacting with double-stranded RNA. These fell into four major groups. One of them is a RNA polymerase, DDX helicase, and DHX helicase. These are some of these are known interactors with double-stranded RNA, others are novel. We saw an RNA processing cluster, a tRNA synthetase cluster, and then a classical double-stranded RNA interacting cluster, including 8R1 and protein kinase R, really kind of telling us that this is working, as well as identifying some new things. And then in our poly-IC sensitization screen, you can see the results of this on the right. We found enriched the loss of pathways that are involved in interferon and double-stranded RNA sensing. So if you lose the capacity to sense interferon, if you lose TLR3 or other double-stranded RNA sensors, the cells were more likely to survive a poly-IC stimulation. In contrast, on the depleted side in blue, you can see a number of genes that seem to sensitize tumor cells to poly-IC double-stranded RNA stress. And when we overlap these, you find a number of genes, but among them, this gene, which is called RBMS1. You said, okay, what is RBMS1? So it turns out that it is an RNA binding motif, single-stranded interacting protein. It's relatively poorly studied. It is known to interact with single-stranded RNA. It has no known double-stranded RNA interaction that's published, although it does have putative effects on PD-L1 mRNA transcript stability. We set out to validate this in tumor cells. We first just did this with shRNAs and said, okay, you see some effect with poly-IC stimulation when you knock it down. Nothing that's earth-shattering, but seems to like there's something there. But then more dramatically, when we stimulated the tumor cells with SLR14, which is a rationally designed RIG-I agonist produced by Anna Pyle here at Yale and validated by Akiko Iwasaki, among others, we saw that CRISPR-mediated knockout cells that lack RBMS1 had a much greater excursion of interferon beta and this other ISG, interferon-stimulated gene, IFIT2. So you were seeing a stronger response to double-stranded RNA stimulation in the RBMS1 knockout cells. We asked where RBMS1 is localizing within the cell and found that it partially co-localizes with double-stranded RNA. So we used our B2 reporter, as well as, in some cases, the rhodamine-labeled poly-IC or the mitochondria, and we looked for co-localization with RBMS1, which was partial. So you don't see total co-localization of double-stranded RNA RBS1, but you do see partial co-localization. Surprisingly, we found that not only was loss of RBMS1 sensitizing and increasing kind of sensing of double-stranded RNA species, but in fact, it seems to be increasing double-stranded RNA abundance as well. So this is qPCR for endogenous retroviruses that are capable of forming double-stranded RNAs. We found in our RBMS1 knockout cells, following poly-IC stimulation, you actually get a greater abundance of these species. And then even more so, a more dramatic increase when you treat them with 5-azacitidine, which is a DNA methyltransferase inhibitor that is capable of de-repressing endogenous retroviruses. We validated the increase in double-stranded RNA using a more traditional double-stranded RNA sensing approach, the J2 antibody, which binds to double-stranded RNA, can be used in fixed cells. And we did this over a variety of tumor cell types, from melanoma, breast cancer, and colorectal cancer in both mouse and human cells. We treated all of these with 5-azacitidine. And in each case, you can see some upregulation of double-stranded RNA, but in the RBMS1 knockout cells, this was always a greater effect. So we're still working on the precise mechanism by which RBMS1 regulates double-stranded RNA abundance and sensing. Two possibilities include, direct kind of binding and protection from degradation, competition with sensors, versus a change in trafficking patterns that leads to the same effects. In either case, we wanted to know, does it matter? So for this, we collaborated with Akiko Iwasaki's lab to ask whether RBMS1 can affect SARS-CoV-2 infection. To do this, we took our tumor cells and we expressed the ACE2 receptor and then worked with the Iwasaki lab to perform SARS-CoV-2 infections. And as you can see in the RBMS1 knockout cells, you get significantly less SARS-CoV-2 infection, suggesting that this increased double-stranded RNA sensing was actually protecting against infection. Conversely, if you overexpress RBMS1 in these cells, you actually get more SARS-CoV-2 infection. Finally, we looked at a human cohort that had RNA sequencing data from blood available in patients who either had more severe COVID infections, meaning hospitalization or death, or less severe infections, meaning they did not have hospitalization or death. Intriguingly, we found that RBMS1 expression was significantly higher in the more severe patients than in the less. We next asked whether RBMS1 affects tumor immunity using MC38 and B16, MC38 colon cancer transplantable models and B16 melanoma models. Indeed, we found that the loss of RBMS1 significantly controlled tumor growth in MC38 and B16 and increased survival, and furthermore, that this appeared to be dependent upon type 1 interferon sensing. That is to say, when we used antibodies to block type 1 interferon receptors, you saw a significantly attenuated effect here. RBMS1 knockout no longer helped control tumor growth as much as it originally had. we looked within the tumor microenvironment of our MC38 tumors. And within those tumors, we found that the RBMS1 knockout tumors had significantly more immune cells, CD8 T cells, CD4 T cells, and NK cells, and greater granzyme B expression in both the NKs and the CD8 T cells, supporting the idea that you are driving improved anti-tumor immunity by the loss of RBMS1. So just to summarize this part of the talk, we validated the B2 reporter as a new tool in mammalian cells for cytosolic double-stranded RNA detection. We identified RBMS1 as a novel regulator of double-stranded RNA sensing and abundance. And we showed that it can impact both viral immunity in the setting of SARS-CoV-2, as well as anti-tumor immunity in what looks like a type 1 interferon-dependent manner. Okay, that was story number one. The next story I'd like to tell also involves the regulation of double-stranded RNA and interferon sensing, but is more translational and comes directly from patients. So in this case, it begins with the development of perturbational single-cell RNA sequencing approaches that we've been applying directly to patient tumor and immune cells. And our motivation here was fundamentally just to do immunology in patient tumors. So, in the lab and mouse models, we can agonize pathways, deplete cell types, kind of manipulate the system and really see how these perturbations shape tumor and immune interactions. But that's more difficult to do in patient tumors because they don't live, the cells don't live for very long in their native proportions once you remove them from the patient. And so the way that we have done this is to take surgical resections that have both tumor and immune cells, to make single cell suspensions and plate them in up to 10 different conditions, 10 so far, though we think in the future we'll be able to do potentially many more, and to stimulate them with different drugs or to manipulate the systems. We run the reactions for only 48 hours because this is the amount of time that we've initially found will support the native proportions of immune cells, particularly myeloid cells. Then we immune enrich and use cell hashing to do single cell RNA sequencing together. And this project has been led by one of our talented medical oncology fellows, Kurt Perry, who will be an instructor in medicine soon, as well as computationally by Yue Fei, and has involved collaboration with our surgical colleagues, Dr. Kelly Olino, as well as Alex Fry, and more recently has been joined by another one of our talented medical oncology fellows, Yunan Ni, as the project has taken a turn towards small cell lung cancer. So one of the virtues of this approach, as I mentioned, is that we can test multiple perturbations. And we selected perturbations around a variety of states of clinical novelty, from standard of care drugs like PD-1 to things that have been tested only in early phase trials or have never been in human trials. These range from cytokines to innate immune agonists. We were particularly interested, as I mentioned before, in double-stranded RNA as an axis for manipulating tumor and immune states. And so we used a lot of double-stranded RNA mimetics, poly-IC, or this novel agonist SLR14, and also we're able to test combinations like interferon beta plus PD-1. And on the right, you can see a batch-corrected kind of dimensionally reduced plot showing Merkel cell carcinoma, melanoma samples, as well as healthy PBMC samples that we've collected thus far. The motivating question for us, as I mentioned, is really what is the role of the antiviral response in shaping patient tumor and immune interactions? And underscoring this, there's been an interest in STING agonists and triggering type 1 interferon for a very long time in the field, yet these drugs have largely failed to translate. And so we wanted to understand, they've been mostly validated in mouse models and then gone into clinical trials. Can we really understand what's happening to the tumor and immune cells when they're treated with these types of drugs, and perhaps identify mechanisms of resistance that are present in human tumors that are not previously observed or appreciated. As a first step, we wanted to understand whether we were capable of detecting standard of care therapy responses, including PD-1. Of course, a lot of the action for PD-1 blockade is in the draining lymph nodes. However, we wanted to know whether we could see transcriptional evidence in our single cell RNA sequencing system for PD-1 responses in tumors. And the correlate for this is work that has been performed by a group in the Netherlands, Daniela Thoman's group, where they've looked at ex vivo tissue slice models and seen that these can be predictive of patient responses. And when we looked, indeed, we can see we can see interferon gamma signatures that upregulate in response to PD-1, suggesting that you actually are detecting PD-1 responses in the setting of our system, as well as cytokine bead array increases so that the cells are making more cytokines following PD-1 blockade in the system that we're using. We next asked whether we were inducing antiviral effects. That was kind of the goal of our study. So we said, how do these different treatments affect the antiviral axis, and particularly the hallmark interferon alpha axis? And what we observed is that basically all of the agents had some effect if you averaged it out over the number of different patient samples and cell types. However, 3 really stood out. The first was interferon beta combined with PD-1, which was the very strongest in inducing type 1 interferon signatures. The next was interferon beta alone. It's a very high dose of interferon, so that's maybe not totally surprising. But the third was actually this drug SLR14 that I mentioned, this novel RIG-I agonist developed by the Pyle and Iwasaki labs. And we were interested, oh, that's not projecting very well. We were intrigued by the strength of response to SLR14. If this looked better, what you'd be able to see is that the variety of cell types we showed before actually have, even at this resolution, I think you could see the red and the black separated. So the red are SLR14 treated and the black are not. And basically what you see is a shift in every single cell type transcriptionally following SLR14. And then on the right, which you can actually read, we show the overlap between interferon beta-induced genes and SLR14-induced genes. And what you see is that SLR14 gives you most of the same genes as interferon beta and an additional gene set as well, including some T-cell and immunity-relevant genes like perforin or CCL5, important chemokines or anti-tumor response genes. Moreover, when we look at just the T cells, and this is using an algorithm developed by the Krishnaswamy lab here at Yale, we see a progression of antiviral states with SLR14 stimulated T cells. In the CD8 T cells, you see this separates out nicely on the Y and X axes with antigen experience and antiviral states. And moreover, that interferon beta lies somewhere between the unstimulated states and SLR14, that SLR14 pushes you further down this differentiation pathway. When we looked to try to understand the specific genes that were mediating this effect, in the CD8 T cells, we saw genes associated with activation, CD69, as well as CXCL13, an important chemokine. In CD4 T cells, lower FOXP3 and lower TOX and PD-1, actually, as well as a more traditional set of interferon-stimulated genes in NK cells and tumor cells. Given these findings, we wanted to see whether SLR14 truly helped to activate T cells, particularly given prior reports that STING agonists, at least at high doses, can kill T cells and actually be counterproductive. And indeed, when we stimulated T cells in the context of healthy donor PBMCs, with SLR14 followed by CD3 and CD28, kind of classical activation, you see an increase in the interferon gamma producing cells following SLR14 and in interferon gamma granzyme B double positive cells, really supporting this idea that SLR14 was activating the T cells in a helpful way. We next sought to understand variations in the patterns of response and resistance among the Merkel cell carcinoma and melanoma tumor samples in our cohort. Here we used an algorithm called MELD, which measures transcriptional response to stimulation using matched treated and untreated samples. And the thing that stood out for us first is just that when you compare the samples derived from melanoma tumor samples to those from Merkel cell carcinoma, that the melanoma samples had a significantly greater response to both SLR14 and interferon beta. We wondered about this. To interrogate these findings, we applied an approach that had recently been developed by the Van Dyke lab here at Yale, and that we'd kind of helped to validate, called Cinema OT. And the way this works broadly is that it uses a mathematical approach called optimal transport matching to separate out confounder from response variation in matched treated and untreated data. And in doing so, can help to isolate kind of features of that response. And when we applied this approach, the gene that came up in our kind of Merkel cell carcinoma non-responders above all others was this gene, MIDKINE, MDK. And accordingly, MIDKINE, or MDK, was higher in Merkel cell carcinoma than in melanoma, and higher in the Merkel cell carcinoma non-responders than it was in the responders. And so we returned to the literature. We said, okay, what does midkine do? I didn't know much about midkine. And it turns out that midkine is a pleiotropic cytokine that has at least 5 different receptors and a number of different effects. However, there were 2 stories in the literature that were particularly relevant to us. The first was one from the Songus lab that had identified midkine as a potential driver of resistance to immunotherapy in melanoma. This was interesting to us because actually in our data, it looked like the expression was much higher in Merkel cell carcinoma than it was in melanoma. But be that as it may, they had shown that maybe there's an effect on tumor-associated macrophage polarization, that you got more suppressive TAMs, basically when you had midkine high tumors. The other story was from Nellie Polyak's group at the Dana-Farber, and it really characterized midkine in the context of aging, as associated with aging, and in the context of breast cancer and tumorigenesis, as a driver of PI3 kinase and mTOR pathways and effects on tumorigenesis. Neither one of these papers had actually studied effects on interferon signaling or in neuroendocrine tumors. So we said, okay, this will be a novel context for thinking about midkine. So what we set out to do was kind of reverse translation and to return back to the bench from these patient samples to ask whether midkine was doing something to suppress interferon signaling. And when we did this, we first realized that midkine is very difficult to work with. It has a short half-life, and it was challenging to do kind of re-stimulation experiments. And so we turned to our CRISPR-based tools. First, validating that the Merkel cell carcinoma cell lines that we had in the lab make significant quantities of midkine, and then using CRISPR and Cas9 to delete midkine so that they no longer do. When we did this and then re-stimulated these Merkel cell carcinoma tumor cell lines with interferon, either interferon gamma or interferon beta, we observed that the midkine knockout tumor cell lines have significantly greater responses to interferon than do the wild types. Right? So this is true in either OASL or ISG15, kind of classical interferon stimulated genes. We asked next, because this affected interferon, whether it was also affecting MHC1 expression, given that MHC1 is a classical interferon stimulated gene. We said, well, why would this matter for tumors? Well, one reason it would matter is that if you don't sense interferon as well, you don't upregulate MHC as well, and that could actually help you in immune evasion. And sure enough, in our tumor cell lines, following deletion of midkine, we saw a greater upregulation of class 1 MHC following interferon stimulation. At this point, we had a different idea. So one of the things that we had noticed in our original single-cell RNA sequencing of Merkel cell carcinoma tumor samples is that some of them, the entire sample failed to respond to interferon or to any of our innate immune agonists. It wasn't just the tumor cells, it was also all of the immune cells in the sample. And so we wondered whether midkine would have similar effects in limiting the expression of class 1 MHC on our immune cells. And sure enough, when we did co-culture of either our midkine knockout or our wild type tumor cells, along with CD14 positive monocytes from healthy donor PBMCs, we found that the midkine knockouts had significantly greater upregulation of class 1 MHC, suggesting that midkine is limiting MHC expression not only on tumor cells, but also on immune cells. We furthermore asked whether there were other effects on the immune cells, looking now at both CD14 positive monocytes and monocyte-derived dendritic cells. And we found that in the midkine knockouts, you get not only more upregulation of MHC1, but also upregulation of co-stimulatory molecules that are important for activating T cells, such as CD86. Given that the effects, that the expression of midkine was so much higher in Merkel cell carcinoma than it was in melanoma for us, we hypothesized that midkine might actually be increased in other neuroendocrine tumors, including small cell lung cancer. We therefore examined midkine expression in bulk RNA-seq data available for melanoma, Merkel cell carcinoma, and small cell lung cancer. And we found, somewhat to our surprise, that in small cell lung cancer, the expression was even higher than it was in Merkel cell carcinoma. Working with Arnaud Auger's lab, we next looked at a cohort of small cell lung cancer single cell RNA sequencing data, which again confirmed high levels of expression by the samples and specific expression to the tumor cells, that it's actually the tumor cells that are making the midkine that's causing immune inhibition here. We then returned to the lab using cell lines that the Auger lab had provided us with and used our CRISPR system to make the knockouts just as we had done in Merkel cell carcinoma. Again, we found that following deletion of midkine, you got a stronger upregulation of class 1 MHC, both with, in this case, and without interferon stimulation. Working with yet another of our awesome clinical oncology fellows, Amin Nassar, we accessed the KERAS data set in small cell lung cancer, which is about 870 patients, a large data set of transcriptionally matched RNA sequencing data, along with clinical treatment outcomes. And separating midkine by median expression, you see in this data set a lower expression of CD8 T cells and M1 macrophages and a higher expression of M2 macrophages when you have midkine high compared to low tumors. So supporting the idea that midkine is holding down productive anti-tumor immunity. And when you look at immunotherapy response, you see a significant increase in response and improvement in event-free survival in the midkine low tumors compared with the midkine high tumors. In fact, if you kind of track it down by quartiles of midkine expression, they go in order from low to high, from longer survival to shorter survival. So putting this part of the talk together, we've used this perturbational single-cell RNA sequencing approach directly from patient tumors to identify midkine as a factor limiting anti-tumor immunity in a variety of neuroendocrine tumor types. We showed that RIG-I agonism can enhance T-cell activation states, but really kind of identified, I would say, a few places at which midkine is breaking tumor immunity cycles. First, at the level of recognition of tumor cells by T cells, because MHC1 is not upregulated efficiently. Second, at the level of interactions with antigen-presenting cells, because antigen-presenting cells are also no longer upregulating MHC efficiently or upregulating co-stimulatory molecules like CD86. So we think that this is, kind of potentially a multimodal breakdown of tumor immunity that's happening. Okay. Since we're doing okay on time, the last story I wanted to talk about is actually the development of, is more therapeutically focused, and is focused on how we make better vaccines and better mRNA vaccines to virus-associated cancers. And in this case, this is a collaboration that we started with Dr. Olino and with Alex Fry, who's a surgical resident again in the lab, appeared in the last story as well. And this grew out of really clinical need. So we see a lot of Merkel cell carcinoma patients. I see Merkel cell carcinoma patients. And this was one of our COVID projects, was its genesis. We were looking at the efficacy of some of the mRNA vaccines in driving effective CD8 T cell responses. And then we looked at the unmet need in Merkel cell carcinoma. So Merkel cell carcinoma is stage for stage the most lethal skin cancer. Melanoma kills more people, but Merkel cell carcinoma kills a greater proportion. And in 80% of cases, it's associated with a virus, the Merkel cell polyoma virus. The standard of care, first line, is checkpoint inhibitors, but they're only really effective in about half of patients, and recurrences are common, probably more common than they are in melanoma and other diseases. And there is no clearly established second line. Immunotherapy combinations are often tried, although they have pretty variable efficacy in different case studies. Chemotherapy works about like it does in small cell lung cancer. There's a response rate, but it's often transient, it comes right back. And so, but we knew that the virus had a few properties that made it an optimal target for mRNA vaccination. Merkel cell polyomavirus, it really works by integrating oncogenic T antigens into the host genome. So it's an endemic virus, 40 to 80% prevalence, but the way that oncogenesis happens, these particular T antigens get stuck into the host genome, a C-terminal helicase gets truncated, and this permits RB1 binding in oncogenesis. One of the important features of the viral T antigens is that they are absolutely required for tumor cell survival. So if you knock down the viral T antigens with an shRNA in vitro or in vivo, the tumor cells die, meaning they cannot escape it. And so we said, strong antigen dependence. For that reason, these antigens are absolutely conserved. And we know that they're highly immunogenic, not just because they're viral antigens, but because when you look at cohorts of patients who have successful PD-1 blockade, you can actually find in the peripheral blood CD8 T cells that are specific for the viral antigens. The viral subtype also does not have a high tumor mutational burden, in contrast to the UV-driven subtype, really supporting the idea that, for the most part, the immune system is targeting the virus. So we know that it is targetable and that it's essential. And we said, wow, it's a little bit like, we almost didn't do it at first because we said it's a little bit like falling off a log that there should be an mRNA vaccine against this thing. That was in a different time period, admittedly. But to develop one, we first had to develop a model. So we took a B16 melanoma model because there is no transplantable, easily accessible Merkel cell carcinoma model. And we expressed the large T antigen. And first we showed that, the tumor cells grow the same. And next, we optimized a vaccine platform, including kind of some engineering, as well as implementing protocols in the lab to do in vitro transcription. And when we did this, we could use our vaccine in either antigen expressing models, that's the B16 LTA, or empty vector matched controls. And as you can see in the antigen expressing models, the vaccine significantly controls tumor growth, that's in red, and improves survival, suggesting that this was in fact, working. Moreover, the vaccine combines cooperatively with PD-1 blockade, as in you get a better response with the two together than you get with either one alone. That's important since PD-1 is the standard of care approach in Merkel cell carcinoma and a place where a lot of these vaccines have been developed. We next asked, though, we realized that our model had heterogeneous expression of the LTA. It was not a single clone model initially, and unlike virus-associated Merkel cell carcinoma, the B16 model does not depend upon LTA expression for survival. We know that because it was surviving fine before we put the antigen in. And so we hypothesized that our responses were actually potentially limited by loss of expression of the LTA protein in our model. And indeed, this turned out to be true. We took day 24 tumors out and examined them. And we saw, first of all, a loss of expression of LTA compared with input control, but actually, strikingly, a greater loss in the vaccine-treated mice than in the placebo-treated mice, suggesting that you are, in fact, getting selection against the LTA-expressing tumors. Importantly, this mechanism, as I mentioned, is not possible for actual Merkel cell carcinoma. And so we made a single cloned model that at least, still may be able to lose expression through epigenetic silencing, but starts out with uniform expression. And in this case, when we combined this model with vaccination and PD-1, saw 100% cure rate, an improvement in the overall response to vaccination, and with PD-1, 100% cures. So great that this works in mouse models. Lots of things work in mouse models that do not translate. Because we treat these patients, we wanted to ask whether we could actually see evidence of vaccine efficacy in patient samples. And to do this, we developed an approach for ex vivo vaccination, where you can take patient blood and use our vaccine or kind of placebo LNPs to expand out patient T cells. Right? And then we can characterize those T cells when re-stimulated either with matched monocyte-derived dendritic cells from the same patient with vaccine or without, or against HLA-matched tumor cells. As we did this, we used a tetramer to track whether we were expanding antigen-specific T cells. And indeed, in 3 separate patients we've done this in, you can see an expansion in tetramer-positive T cells over time. For those same patients, we asked whether following re-stimulation with HLA-matched Merkel cell carcinoma tumor cells, you could see an increase in functional properties like interferon gamma secretion or tumor cell killing. And indeed, the LTA vaccine expanded T cells had increased interferon gamma secretion following re-stimulation compared with controls. And they also had improved killing of the matched Merkel cell carcinoma tumor cell lines, suggesting that we were expanding functional T cells here. For one patient, we had access not just to HLA-matched tumor cells, but primary matched tumor cells. And so for this patient, we expanded the T cells for 25 days using their own monocyte-derived dendritic cells in our vaccine or placebo control. And then we re-stimulated them with the primary tumor cells overnight. And when we did this, we found a few things. We performed single-cell RNA sequencing with matched TCR sequencing. Using this approach, you can see the outgrowth of TCR clonotypes that are specific to the vaccine-stimulated condition. These are putatively vaccine-specific TCRs. And you also see a variety of transcriptional programs, including cytotoxicity, interferon, and chemokine expression and proliferation that are increased in the vaccine-expanded compared with the placebo control-expanded tumor cells. This is the same data shown another way, where we've highlighted the cytotoxic effector cluster and the proliferating cluster. And you can see comparing the LTA vaccine expanded T cells on the right with the placebo expanded T cells on the left, that you have greater cytotoxic and proliferative kind of density, but also this is kind of going down top to bottom, increasing antigenic stimulation, right, with either placebo dendritic cells, with tumor cells, or with LTA vaccine-loaded dendritic cells, that you get antigen-specific enrichment within the proliferation category. So you really are driving, antigen-specific proliferation. And so we said this is great. It looks like there's a path forward for this vaccine translationally, but we realized at the same time that just dropping in antigen and LNPs, kind of innate stimulation plus antigen, may not be the optimal way to expand the types of CD8 T cells that we want to expand, right? Tumor vaccines have been around for a long time and haven't had as much success as we would like thus far. And we realized also that we were working in mRNA, which is this kind of modality where you can encode any kind of signal that you want. And so we said, well, maybe if we start to coordinate multiple signals together, if we create adjuvants and multiple signals in series, or that we can actually improve various aspects synthetically of the T cell expansion that we're trying to achieve. In theory, this could range from optimizing pattern recognition receptor agonism to attracting dendritic cells and activating them, building supporting stroma, or just expanding the right types of phenotypes of T cells. We said, wow, there's a whole palette of things to play with here. So we wondered where to start. And where we ended up starting was IL-7, and this was for a few reasons. First, because IL-7 has a good basis in the literature for potentially combining well with vaccines. There's work by TAC-MAC that goes back to 2009 or so. But second, due to our own analysis of Merkel cell carcinoma patient data. So we accessed a cohort of patients and found that CD8 T cell proportion, as well as the stem-like CD8 T cells that we think are important for mediating immunotherapy responses, correlated very strongly with IL-7, maybe not super surprising, but also that some of the translational data in the Merkel cell carcinoma field has pointed to memory T cells as being one of the strongest associations with immunotherapy response. And so we built in a co-encoded antigen plus IL-7 vaccine. And you can see that's kind of in the red versus the pink, that you do improve kind of control of tumors as well as kind of survival of the mice. And more than that, you also see an increase in dextramer. These are dextramer instead of tetramer, but are antigen-specific CD8 T cells. So we found an epitope that is expressed on mouse class 1 MHC and used a dextramer to detect antigen-specific T cells for it. That is increased as well with LTA plus IL-7 vaccines compared with IL-7 alone. Looking within the spleens of vaccinated mice, you see an increase in memory phenotype CD8 T cells, and strikingly, only in response to LTA plus IL-7 vaccines did we see an increase in memory phenotype antigen-specific T cells. We didn't really see that with the regular vaccine in the absence of IL-7. So we think that this is actually a better way to build memory T cell responses. So summarizing this last part of the talk, Merkel cell polyomavirus MCC we think is an optimal vaccine target. It's a strong universal antigen that has absolute dependency for tumor cell survival. mRNA vaccination selects against antigen expression. Vaccination enhances immunity in mouse models and ex vivo patient samples. And finally, that co-encoding IL-7 improves vaccine responses, right? And we think that IL-7 is sort of just at the beginning of things that we should be studying, even if just focused on T cell memory. There are a number of other combinations that we're actively testing in the lab now. So just to wrap up, some ongoing work in future directions. At a very high level, for perturbational single-cell RNA sequencing in the PERCEPT project, we want to apply this to other therapeutic settings. We are working with the Van Dyke Lab, who have developed large language models capable of analyzing this data and are trying to integrate some of our perturbational studies with their large language models, the cell-to-sentence model. We're also trying to scale the number of perturbations. We think you might be able to scale this up significantly using different types of single-cell RNA sequencing approaches. For the mRNA therapies, co-encoded combinations of multiple different cytokines, but even more than that, other signals for dendritic cells, signals for building tertiary lymphoid structures. There's a lot of kind of ongoing interest in the types of directions you could push this work now that you have synthetic control over the other signals in the immune microenvironment. And then for the double-stranded RNA reporter, we really want to understand the mechanism of RBMS1, as well as the underlying condensate biology that we've observed. Some other ongoing areas of studies that I didn't have time to talk about today include mechanisms of enhanced dendritic cell function in ADAR1 null tumors, potentially mechanisms by which those tumors are directly priming CD8 T cells, feed-forward and self-propagating inflammatory signaling networks. We think that this may have to do with trained immunity, particularly in head and neck cancers, the determinants of response to oncolytic viruses, and finally, a set of novel CRISPR-based transcriptional sensors and cell programming tools. So I will wrap up there, but I wanted to thank everybody in the lab who has driven this work forward, as well as our collaborators, our funders, and of course, patients and their families. And with that, I thank you and we'll take any questions.
Jeff, that was terrific. So excited about the small cell work. And I guess I have a couple of questions about that. One is, which we can continue to talk about. Have you dived in a little bit deeper to look at responses to immunotherapy in available data sets that have MIDKIND expression, if that correlates? And then the second question is just thinking a little bit more therapeutically, what would be the angle there?
Yeah, no, great, thank you. And sorry, I was a little worried about time and I may have blown through a little too fast, but we did actually look to the first one. We did actually look in the KERAS data set for correlations with MIDKIND expression and immunotherapy response. These were patients, again, about 870 patients that were treated with any type of immunotherapy. So, and that's kind of on the right here. And if you just split it by quartiles of MIDKIND expression, we saw improved survival in the lowest quartile of expressors, and the quartiles counted like directly up to the worst survival in the highest MIDKIND expressors. And in terms of, sorry, the second question,
which I think is terrific, and I was thinking of even diving deeper in terms of looking at specific data sets with a response to immune therapy, for example, resistance and so forth.
Yeah. So you're thinking about like mechanisms by which you see those correlations? Yeah, no, that would be exciting. We'd love to think about ways to do that. Did I answer both your questions though? I think you had a second.
And then the therapeutically?
Oh, therapeutically, how to carry this forward. Yeah, that's a great question. We think that there should be an antibody to this. The Songus lab, after they identified MIDKIND as a potential melanoma anti-tumor immune mechanism, started developing inhibitors. There's an inhibitor that we purchased. We've had trouble getting it to work very effectively in the lab. I think because it's a cytokine, it's a fairly obvious antibody target in my mind. There would maybe be even knock-on, ways to think about the role of MIDKIND in aging. So we're, interested in doing that and potentially working with partners to do it.
Very cool. Thanks.
Katie, do you want to take the microphone?
Thank you. That was great. I have a MIDKIND question as well. I was wondering a little bit about what you're thinking in terms of the molecular mechanism. So it's a cytokine, and you showed a lot of effects on different cells. Just wondering what your thoughts are about that.
This is a great question, and we're not all the way there yet. And we thought we were, so we were trying to track down the receptors, and we were looking at particularly Notch2 signaling and LRP1 signaling. We used what are some of the available tools to block these pathways and have frankly seen kind of variable results with those assays. So I can't tell you today that it's LRP1 or that it's Notch2. Those were Notch2 receptors, those were the ones we were thinking about. The other thing I will say is I think it's not as simple as interferon. So I think that if we did RNA-seq of some of our knockout cells, versus our wild types. And we saw, yes, interferon signaling pathways that were altered, but really a lot of NF-kappa that were altered as well. And so we think that this may be, through one of these receptors or combination of these receptors, we haven't yet been able to figure out yet, that you may be altering the NF-kappa B set, and in turn, indirectly affecting interferon responses. There's at least one possibility, but more to come.
Thank you.
So I'm going to ask you a question about your RMBS1 project.
Yeah.
Where you're taking that one?
Yeah. So, it's translationally or mechanistically or both. So mechanistically, I think it's going to be really understanding, is it a direct interaction with double-stranded RNA or not? And is it changing the trafficking of double-stranded RNA so that it co-localizes or not with various double-stranded RNA sensors? Those are some of the things I think we need to do. Translationally, I think we're not sure yet. So, I will fully admit the phenotype, while clearly there and clearly present, isn't as strong as the ADAR phenotype, right? And it's not clear to me. It also depends how it's working as to whether it'll be a very easy drug target or not. It's kind of a floppy, maybe non-enzymatic molecule. And so it's not clear that it would be a great small molecule drug target at present.
Okay, thank you. Any other questions? In that case, Jeff, thank you so much for a wonderful talk. It was a real tour de force. You're doing a hell of a lot, and we appreciate it all.
Thank you.